Hi, I'm Spencer Watson. I'm a cancer biologist at the Agora Cancer Research Center, and I study the tumor immune microenvironment and ways that this can cause therapeutic resistance, primarily using high-dimensional imaging. I think in the next 10 years, the biggest advances will be integrating in all of our current omic approaches like single-cell RNA-seq with spatial biology so that we can find new emergent features and potentially new therapeutic targets. I think I first saw a demo at a ISREC conference here in Lausanne, Switzerland. I think we had a few interests in it. Primarily, we wanted to be able to test antibodies and test tissue samples very rapidly. But then there was also the possibility of using the LabSat for multiplexed imaging. Yeah, the main challenges we had with developing multiplex stainings before we had the LabSat was that it was very labor intensive because we were doing everything manually, and just that it would take more than a day to get any sort of results back, and we typically need to get these results a lot faster. LabSat helped to address the challenges we were facing primarily by bringing in this automation, which included a lot of ease of use, which was especially helpful because then we could train people on this very quickly, very easily, and then get our results back in just a few hours rather than a few days, while still getting really good quality. Our current workflow using the LabSat is primarily for antibody validation. We run very large, high-dimensional imaging experiments that require very large antibody panels that all need validation on the tissue of interest. So this would take us an extremely long amount of time to do in any sort of manual way. So with the LabSat, we're able to go through all of our antibody panels and validate these all really quickly. So our plans for using the LabSat for future projects are also for antibody validation for these very large antibody sets, but we also get a lot of patient samples in from neighboring hospital and our collaborators, and we really need to get results from these samples very quickly so that we can tell if they can be included in one study or another, and that's where the LabSat becomes very useful.